following is a conversation with Professor Julian Marchesi, Professor of Digestive Diseases at Imperial College London. Over his 22-year career, Professor Marchesi has authored over 190 peer-reviewed publications. He currently sits as chair of the British Society of Gastroenterology's Gut Microbiota for Health expert panel, and also chaired the Microbiology Society's Unlocking the Microbiome publication and working group. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. So, not Inside Matters, Julian, but I'll tell you something, right? So, people find it very hard to stay in silence. And I read this article that's talking about an experiment where they gave young men a choice. And the choice was you can either sit in silence for like 20 minutes or you can electrocute yourself for 20 minutes. And guess what they picked?
Electrocution?
Yeah.
I could do the silence.
Of course.
I can do the silence.
You ever heard of these silent retreats?
I've always wondered if it'd be, well, I have, and I always wonder if those isolation pods.
Yeah, the isolation chambers.
Where you just actually cut out all stimulation.
Yeah.
I don't know how long. I mean, some people say you can last 10 minutes. Some people just hate it. They freak out.
Right.
And the other one is anechoic chambers, which are totally silent.
Wow.
And a lot of people say that they can't cope with that because they just start hallucinating, oral hallucinations. When all noise is cut out, you can start hearing your heart.
Yeah, right. It's a bit freaky,
and then you start hearing other noises. And some people say, no, get me out. 10 minutes, 20 minutes, people start going really freaked out.
Wow. Well, the silent retreat is something I am going to do. I went to a Buddhist monastery last weekend called Samiling. It's one of the largest in Europe. It's in Dumfries, south of Scotland.
Okay. I wouldn't have put one there.
It's amazing. It's amazing. Yeah. So I went just to check it out because I'm all about just trying new things, anything that could be related to wellness, health, fitness, or whatever. I'm kind of keen. Mindfulness, So they do a day, 2 days, 7 days, 10 days, I think. Maybe not specifically there, but some people do a 10-day silent retreat. And whilst on the face of it, that might sound quite like exotic and fun. That's quite a long time to not speak to someone. Do you know what I mean? Yeah. Like you're sort of like, oh, what do I do?
And no devices.
Oh, no devices. No.
No Wi-Fi.
No Wi-Fi. Nothing.
Just silence.
Just you.
Well,
and your
not
thoughts.
silence. I mean, you've got the environment around you.
And they do meditations.
Yeah.
Maybe there's a bit of yoga. But the yoga, the breathing, the cold water, and the sauna, I think, really changed my life in a positive way.
Cold water. Okay.
Ice cold water. There's a place beside, and I don't know what the effect is in the microbiome. It seems as though I read an article every day, and it's like this benefits your bacteria.
I know.
Exercise benefits your bacteria. I shared it because it's BBC, and I like hyping things up. But I'm like,
everything can help you. Look, we're here to discuss.
We are,
microbiomes,
and
they've had a bad press. I think they've had a bad press. And they've had a bad press because a lot of people have been pushing them. And it's an easy push. It's not like trying to push graphene or artificial intelligence because a lot of people don't get it. You ask somebody, what's graphene? I have no idea what graphene is. 99.9% of people, what's artificial intelligence? Something that's artificial and intelligent.
One of my biggest beefs with AI is I keep asking people what it is, and they all tell me something different.
Yeah. Half the time it's just machine learning.
So it's just machine.
And it's not. I did argue. I wouldn't say argued. I think I did try to email one of the leading lights in this. I can't remember his name now, but he wrote a book called Life 3.0, which, if you're into artificial intelligence, is a great read.
Right.
Saying, should we call it synthetic intelligence? Okay, SI, and call artificial intelligence augmented intelligence.
Interesting.
Because all the things that they seem to be doing are augmenting what we do. So just like a JCB's augmented muscle. Okay, you wouldn't call it an artificial human.
Right.
It's augmenting. It's augmenting what we can do. I can pick up a small stone. A JCB can pick up a very large stone and move lots more earth than me. And a lot of the methods that you see in artificial intelligence are pattern recognition, which humans do very well. It's one of our evolutionary traits, so we can see patterns, and that helps us discern maybe whether there's a predator in a hedge. So we can see the pattern in there and we can get out of it. But then what it also leads to means we see patterns where there aren't patterns. You look at a cloud, you can see a leopard in the clouds, but there's obviously no leopards up there. It's just a shape.
Right.
And a lot of artificial intelligence things or models that you see, a lot of them are just pattern recognition, but done much better because you can put in a lot more data into a computer and it can sift through all that data much better than human mind can find and find a pattern. Whether it's meaningful or not is another thing. So I try to push for this. Let's keep AI as augmented intelligence and real intelligence, which is something we've made, which is synthetic, we've synthesized. Let's call it SI. Totally blanked me, never emailed me back. So
Fascinating.
I went with that. If it ever comes up in the future, you heard it here first.
Here we go.
but
Prof Marchesi on Inside Matters.
Yeah, but getting back to
the microbiome. And I'll make a note, we should definitely talk about AI and machine learning in the context of the microbiome.
Oh, it's everywhere.
I'll make a note. Yeah.
Anything that's got lots of variables, that's too complex. If it's got more than 2 variables or 3 variables, the human mind sort of implodes, isn't it? You've seen somebody walking down the street in a 3-dimensional world trying to text and then walk into a lamppost. That's somebody coping with 3 variables, which is 3-dimensional, well, maybe more than that.
Or just like me this morning, I walked right past Costa Coffee and
simple
totally
thing,
missed it.
Because your mind's somewhere else. But so anything that's multivariate or lots of variables,
it could be
machine learning helps.
So let's bring us back to the microbiomes and the bad press. And
so
I'm keen to learn about your journey through the microbiome, like how it started and how it's changed and where we're at now.
Yeah. So I got into this area 21 years ago. So I trained up as a biochemist, and then I got into microbiology, and then I got into environmental microbiology. And the buzz then was, we're going to understand the environment, and we understand that the microbes in the environment are a key player. They're foundational to those environments. Lots of different biogeochemical pathways: carbon, iron, phosphorus, sulfur, nitrogen. All those cycles, which are fundamental to life on Earth, have microbes somewhere in that cycle. And if you mess about with those microbes in that cycle, you will affect the cycle. You could close it down if it's a nitrogen cycle. And we won't know about the impact of that until maybe 20, 30 years into the future, where we realise that sort of ammonia is not being made from nitrogen, plants are dying, and we've done something bad. And so there was a big push to understand the microbes and their roles and the ecosystem functions that they maintained. But the big problem was we were really bad at growing bacteria from the environment. It was this like dirty little secret that microbiologists had. If you took soil, we kept looking at it and thinking, well, we know there are lots of microbes in there, but we can only grow 1% of them. So 99% of the microbes in that system are just dark matter to us. We don't know, they're there, they're important, but we can't pull them out. We can't investigate them in isolation. We can't get at their genomes. We can't understand their biochemistry. We can measure big biochemical changes, sort of changes in chemicals moving through soil. So you can put some glucose into soil, sugar, and you can see carbon dioxide being made. You can see other molecules being made. But who's making it? How are they making it? Who, is it several organisms feeding off different chemicals, cooperating with each other? No idea. And that's a big problem if you're trying to describe an ecosystem, and you can't describe the main players in that ecosystem. So what a lot of, and it wasn't, I helped add to this, but wouldn't say I pioneered this, but a lot of clever microbiologists realized that you could use DNA-based approaches. And this was going back to people like Linus Pauling, who got a Nobel Prize for understanding that DNA could be used from an organism to understand its evolution. It's a fundamental aspect of understanding now. We take it for granted. We can use DNA to understand evolution of organisms. But back in the 60s, that was a big leap to realize that DNA doesn't just only contain the information to make an organism, it contains information on the natural history of that organism, where it's come from, what it's related to, who its neighbour is, in the tree of life. And this was big because at the time we couldn't build a tree of life of bacteria.
And that's because it was too hard to culture them.
Not just that, it's because they all look the same. If you look at the tree of life of big animals, you can see, well, there's a 4-legged animal over there. I'll put in a group of other 4-legged animals. There's animals there who walk on 2 legs. I'll put those in a group. Okay, that's a reptile. I'll put in the group of reptiles. That gives birth to a living organism. That's a mammal. I'll put him in a mammal. So for big animals, morphology and features they have and things they do are easy to start clumping them together and say, they must be related to each other. Apes are related to us. Cows are related to horses somewhere because they're 4-legged. Ruminants are related to other ruminants, and so forth. But if you look at microbes, they're just spheres, sausages, curved sausages, chains of spheres, and that's it. And yet you'll find some microbes that are so different to another microbe, but morphologically, their shape, they're identical. They're just spheres.
Right.
But one could live at the bottom of the ocean, 500 meters below the seafloor, take sulfur as its energy source and turn it into other sulfur forms and cope with high pressure, and it looks like a spiral piece of pasta. You can find another one which lives in fresh water, doesn't need oxygen, where the other one doesn't need oxygen. So totally different metabolically, totally different genetically, and yet you look under the microscope and I say, well, I can't tell the two apart. So this was a real problem for us. We never had a tree of life for bacteria that allowed us to say, if I pull a new bacterium out, I can put it into my tree of life and show actually where it is related to every other organism in that tree of life. And people like Linus Pauling and then others came along. Carl Woese was the other one. Came along and realised that, well, you can use DNA, you can compare DNA from organisms, and if that DNA is identical, chances are that those organisms are identical. And as that DNA becomes less and less identical, it means that those organisms are less and less identical, and they've also evolved different pathways.
Right. And right now we know today that the genomic potential of the microbiome is orders of magnitude greater than that of its host.
Yeah.
Right?
Yeah.
Did we know that then? Or was it hypothesized?
No, it wasn't known then. It wasn't even hypothesized. But what we did know was that bacteria, the way they adapt, they don't change their shape, they change their genomes. And they share DNA. And so what we knew was genetically, bacteria were much more diverse anyway than mammals. Mammals are pretty boring genetically. You know, that's why we can do experiments on mice. Because genetically, they have a lot of the pathways we have. So, you know, we haven't evolved that far from a mouse. Or we haven't evolved that far from a horse. And so if you look at genes in mammals, we all more or less all share the same genes in mammals. There are nuances here and there. And so we knew from early on that bacteria were really good at adapting to environments by changing their DNA because they grow quickly, they evolve quickly, swapping their DNA. And that's why we have this antibiotic resistance pandemic hurtling down towards us, because they share these genes that allow them to be antibiotic resistant. And so what we did in the environmental setting was we said, right, we can't grow them, but we can extract all the DNA from a system. And with sequences,
right,
the early sequences were slow, so we did things on a smaller scale compared to today.
How slow? Like, what was the process?
If you were sequencing 96 pieces of DNA, that was considered a big project.
Wow.
And so what we have to do is you take your DNA, you break it up, you put it into another piece of DNA, which you could then put into the workhorse of the laboratory, which was Escherichia coli. And then you would pick off your Escherichia coli, re-purify the DNA, and send it off for sequencing. So picking off 96 colonies and purifying them, send it off. And it wasn't cheap. You know, it wasn't cheap compared to sequencing today. The price of sequencing per base, per ACG&T, has dropped phenomenally. It's gone through the floor. You know, we're down to 0.0001 pence.
And that's because of the engineering and the technology?
That's because of the Archon X Prize, which pushed people to... So the Archon X Prize, so there were two of them. One of them was to get people into space cheaply. And I think it might have been the second one, was to get either a $10 or $100 human genome. And the idea was...
Wow.
Precision medicine.
Big goals.
Big goals. So, and actually,
really
I think
goals.
it was only $1 million or $10 million prize. So that was it was a nice prize, but the cost of actually making these machines would have outweighed the prize. It was the kudos of being the first company who could say, we can actually sequence a human genome for $10. So the idea was democratize it and put in a GP primary healthcare. You know, somebody would come in, the idea was bit of blood, there's your genome, and the whole personalised precision genomic medicine would kick in. Okay, it never happened.
So people have been talking about that grand vision of, you come to your GP, give a bit of this, get it all profiled, they'll tell you everything.
Yeah.
That people have had that vision for quite a while, right? And
since, was it 2000 when the human genome was done? It was going to revolutionise. And then what we found was, we haven't got that many genes, and we've got a lot of DNA that's in there doing nothing, we thought.
And the... often overlooked but hugely important element of the microbes, which influence gene expression.
Which was never even considered in the Human Genome Project.
So at that point in time then, no one was thinking about applying, there were a couple of people, but it wasn't a big thing insofar as everyone was talking about applying the tech to the microbes.
We never even considered that. Like most things, we stumbled onto it realising, look, this tech was developed to try to do human genomes. At the time, there was a lot of sequencing capacity because the Human Genome projects, they were all around the world. global. And when they sort of stopped doing that and they sort of produced the first draft, it was like, what are we going to do with all this sequencing capacity?
A huge book, wasn't it? That huge thing.
It was just everyone. So then people realised, well, okay, we can start using some of this. And then the second generation sequencers came. Your Roches and your Illuminas and your Iron Torrent, which Roche and Iron Torrent have disappeared now. Illumina won the battle to start off with. These came online. And as an environmental microbiologist, I was working in this area and I realised, look, if I need a career, I've got to change tack. Because this area that I'm working in is densely populated with very good scientists. And finding, like any scientist, you're like an artist. You need to find your creative canvas. You need to find your unique selling point. It's like any artist. Nobody wants to see somebody just producing Van Goghs or producing Monets. You need that unique thing. And as a scientist, it's no different. Getting back to why I moved in, it was a case of I had this toolkit that I'd helped develop or use to look at environmental situations. And at the end of the day, the gut is an environment.
And the
full
toolkit,
of microbes.
Right. And the toolkit then was the second generation sequencers.
It wasn't at the time, it was just DNA-based approaches.
Okay.
It was what we called culture-independent approaches. So it was always this thing about you culture organisms, you miss the majority, and that mindset was carried over into the gut. And so we said, well what can we get? Stool we can get easily, relatively easily.
Right.
50% of stool is bacterial. Not many people think about that, but when they flush down their stool, 50% by weight is bacterial. That's a hell of a lot of bacteria. Really is. You'd struggle to produce grams of bacteria.
Is that right? It's 50%?
50% of stool material is bacterial biomass. It's a lot.
Every day.
Every day.
And we shall talk about intestinal microbiota transfer and all that later, but that's
every day.
Incredible fact.
It's a lot of microbes.
Incredible.
And so we always had a ready source of bacteria and a ready source of, and we could extract the DNA. We'd learned to extract DNA from dirty samples, in other words, soil, because there's lots of material in there that can contaminate your DNA when you're extracting it and can mess up downstream processes. It'll just stop things from working, like DNA amplification or cloning. So we'd solved that for soil and all these different wastewater treatment plants. So we had the toolkit, and the question is, well, what's actually happening in the gut? Because a lot of people, if you take a gut sample and put it on a plate, what you grow are the fast-growing organisms, the weeds, as we call them. You grow E. coli, Escherichia coli. So people started thinking, well, the large intestine is rich in E. coli. It's not.
That was the working hypothesis then.
It was, and a lot of the working hypothesis in microbiology were driven by culture-based approaches. What can you grow on agar? It's an old-fashioned method. It goes back to Robert Koch, Fanny Hess, who people have forgotten about. She was his technician who gave him the idea to move from gelatin, which was very difficult to use to solidify your media, your growth media, to agar. She's not in the history books, but she was instrumental to allowing us to culture. But everything was driven by that, the culture axiom.
If somebody was to gift the world a book with the exact recipe for each particular plate that promoted the growth of a species. So we knew it. We knew how to culture them. Would we use culture all the time because it gives us an advantage, or would we still be moving towards the approaches you're talking about now?
It would be logistically impossible.
It's the logistical element.
It's a logistical element. I mean, you think, for a fecal sample, you might have 160 species in there.
Right.
Okay. In one person's gut. So you need 160. Well, you wouldn't need 100. You need, taking a step back, we think there are about 1,000 different species in if we pulled everybody's guts in the world and analysed it, we think total is 1,000 species. So if you go into somebody's gut and you don't know what's in there.
Hold on a second. Can I just, so if you, the thought experiment there is you collect a stool sample from every person on the planet.
Analyse it.
Pull it together in some sort of master cell bank thing. And then sample the heck out of it. It'd be a very big thing. And each sample, let's just say, would be representative. And you're saying there'd be around about 1,000, give or take, yeah? Does this include the Amazonian tribes who may have much more?
No, I think that there are only certain organisms that have adapted to live in the gut.
Okay.
Look, when I say 1,000, I wouldn't put my mortgage on it. It's not bang on 1,000.
But it's that order of magnitude.
It's that order of magnitude. Right. The total diversity is covered by 1,000 types of organisms. But what you do is when you go to an individual, they may, out of that 1,000, they may have assembled 160 odd in their gut. Your gut will have 160, will share some, in terms of species names, but then you'll have your own unique ones that you've picked up over life from your natural history. So if I came in with a culture-based approach, what I'd have to come in, because it's agnostic, I don't know what's in your gut, I have to come in with media for each of those 1,000 bacteria to start off with, because I don't know what I'm going to be pulling out.
Okay, I see what you're saying.
And so if you think then it's 1,000 different media, and each sample needs at least 6 or 7 plates, 7,000 plates, and you've got to grow them anaerobically, in other words, the absence of oxygen, just for one person. It's just, that's a lot of plates to pour.
Right.
and
when you put it like that, if you're doing some sort of experiment where you're looking at multiple different people to compare, for example, people with a disease or without a disease, then you can just multiply your 7,000 by 100, and suddenly... And if you want to do it over time, however many time points, and suddenly you've got a very large room filled with a lot of plates. Right. It's just not practical. And that also assumes that in that thought experiment that we actually know the requirements. And it sounds like we don't. Even today, fast forward all those... We still don't.
We still don't.
Right.
A lot of organisms we just don't know. And unfortunately, a lot of organisms don't grow in isolation. They grow as consortia. They grow together. Certain organisms will feed in other organisms. So if you try to split them apart, one organism may grow, but the other one may never grow.
Oh, interesting.
Because it requires a chemical from organism A or a metabolite that you can't even consider.
Wow.
And so it's impractical. So that's why another thing was, it was pragmatic. We turned to the DNA-based approaches. So what the DNA-based approaches have now allowed us to do is quickly create inventories of people's stool samples.
Right.
Or whatever sample you want to look at. So it could be a skin swab, could be a nasal swab, could be a vaginal swab, whatever it is. Could be sputum, could be something from the lungs, whatever it is. And if there's bacteria in it, even very low biomass bacteria, when we're saying hundreds of bacteria, we will be able to pull them out and then get to a point where we can either tell you, we'll tell you who's there. Most of the time we'll tell you some idea of its functions. So we can say, look, this organism has these genes, and we predict that these genes can do X, Y, and Z. But there's a lot of dark matter in there. And what I mean by that is, if you look at the tree of bacterial life, I think maybe it's 120 large groups in there now. And what I mean by large groups is phylum. So a phylum is a family of organisms that share characteristics in terms of their DNA similarity, and we just clump them together. In microbiology, we like to cluster things. It makes things easier to study.
And they're at the top of the tree? So
you go sort of kingdom, which is bacteria, archaea, which are these old organisms, eukarya, which we're in.
Right.
That is, so the kingdom of the eukarya has all the organisms you can think of that we're used to, that aren't microscopic. Well, it's even got the microscopic organisms like your parasites and things like that. Anything that's usually multicellular, not always, fits into the eukarya. So parasites, plants, everything fits into there. That's a kingdom.
Got it.
And then you come down a level, which is a phyla. So these are big families of organisms. But the problem is, a lot of them still don't have any cultured organisms as representative. So all they're based on is DNA sequence, a short DNA sequence. So we have lots of families that we see, some of them in the gut, that just have nothing that's an organism you could point at in a culture collection. And what I mean by culture collection is where somebody's grown the organism to purity, so they know there's only one organism in that culture, that growth. They've characterized it, and then they've given it to an American culture collection, the German culture collection. Okay. And so basically what that allows you to do as a microbiologist or scientist is go back and say, can I have that organism? I'll pay you £60 for it, because that's the idea. I know exactly what that organism is being described as, and it'll be a representative of that family. And so when we do start looking at samples, we can say, there's the organism there. Okay, it's a DNA sequence. However, nobody's ever grown this in the laboratory.
Right.
And so I can't tell you much more than saying it's related to that one over there. That one has a representative in the culture collection, and that one does X, Y, and Z. It can take sulfur, it can make methane. So maybe by the fact that it's a neighbour, this one might be able to do that. But unless you can pull it out as a pure culture, you're never going to be able to answer that.
Interesting. So there's still a really important role to the culture.
It is.
It's fundamental to allowing us to phenotypically...
characterize. Exactly. Yeah. And also it closes a circle. It allows you to take an organism out and test a hypothesis. You could put it into a mouse model. Without the actual physical organism, there's nothing to put into a mouse model.
Of course. You can't just put the DNA in.
You can't put the DNA in unless you think that DNA is important. You can put into another organism, and get it to read the DNA, get it to understand the functions and to produce the functions, the phenotype. Sometimes you can do that, but that's hit and miss. That's a real gamble if it's going to work.
Right.
And so basically, we're at the stage now. We've got this great toolkit. We've got these great sequences which we hadn't anticipated and weren't designed for what we're using them for. They were designed for the human genome, to go into your GP and get your genome done and saying your P450 cytochrome means you're not going to react to this drug, or you don't have the right receptors for this drug. Yeah. And we've hijacked that to be able to sequence lots and lots of DNA cheaply from as many samples, and then use that information and the bioinformatics, the computational tools, to say that's what's in your sample, that's how abundant it is, these are the functions it's making. Okay, so that's one omic. That's your DNA-based omics.
But do you not need, like, if you're going to match it up to a sequence you already have, do you not need the pure culture at some point?
What you do is you.
So how do you know what it does if you've never done a pure culture?
Exactly. You don't. You can try and infer it.
Okay. Based on similarity of
based
gene
on
sequence.
Similarities to neighbours which have been characterised.
Right.
Or if it hasn't. In fact, you're not even allowed to give it a bacterial name if there's nothing there. That's being deposited in a culture collection. Where somebody says, "Here's an organism." This is because there are rules for naming things. Like I can't name a bacterium after myself. I'm not allowed to.
There's some people who have name. So Harry Flint, for example, has Flintibacter.
Yeah, no, but that could be one of his. If you work for me, you could name a bacterium after me. I'm not allowed to name my own bacterium after me. I can't call my name Marchesi. But
that's good then, because it would prevent people from just
with
going
egos, just going out for it, naming every. But some people will honour other people by naming a bacterium after them.
Right, I see. I thought it was bestowed upon them from some society or something like that.
Joel Doré there's a Doria.
Okay, right.
Michael Blout is a Blautia. So people have named genera.
Right.
But nobody thought I'm worthy of.
Right. I'll need to have a chat with Ben Mullish then and give him a nudge. I
keep saying to the lab, well, if you find a new organism, my surname's not easy to pronounce, but we can work on it.
Marchesi, I don't know what would we call it. Marchesi.
But you don't find much. It's Marchesi.
Yeah.
And so it's just like, okay, anyway, it's never going to happen. But so what we then thought about was, well, look, that's one omic. Okay, it's an omic. We call it metagenomics or metataxonomics. We have an omic. Everyone loves an omic term,
right?
And that gives you limited amount of information. But in the human gut, what we're really interested in is what they do and how they communicate with the host. And so that's where we have metabomics and metabolomics. So these omics now, then we sort of stepped up and said, well, look, they make a load of metabolites, a load of chemicals.
Before we jump into the other omics, how did the metagenomics, the things we've just been talking about in terms of the DNA extraction, how did they change the field during your initial years? So
this is where we got to the stage that we realised that there's 150 times more genes in the gut than there are in a human genome. Because they created gene catalogues, and you're suddenly looking and thinking, in one person's gut, there are a lot of genes that we can identify. Wow. We don't know what a lot of them do. Maybe 60-70% of them are just unknown, but we can see it looks like a gene. It has a start part, it has an end part, and it's about the right length to be a gene and make a protein.
Wow.
And it makes this protein, we can translate. We have the tools that can take DNA and turn it into the amino acid sequence. And we say, right, okay, it looks all right. We can try to fold it up and see what it looks like. And then we hit a brick wall.
Must have been such an exciting time to realise that there was such diversity of genomic potential in there. But that's people microbes. Not going a bit mad, going, this is...
it was, but it's just a case of, what do we do? What can we do with it? Because with a lot of these microbes, they evolve genes and we don't know their functions. You look at E. coli, even 40% of E. coli's genome, we don't know what it does.
Really?
And that's the best studied organism on Earth. Yeah, people knock the gene out. It doesn't seem to have an effect on growth, but you can find that gene in many E. coli. So it's over evolutionary history, it's been maintained. So we assume, maybe naively, that it's important to the organism, but we can't understand or predict what it does. And so with metagenomes, we get a lot of genes that are the housekeeping genes, as we call them. Those are the genes that every organism needs just to to survive. Genes for DNA replication, making energy, the things that every bacterium has, even our mitochondria have it.
Like the 16SR RNA.
All the things, for making proteins. They're boring because every organism, it's not the interesting thing about them.
Right.
And so when you do metagenomics, a lot of the genes are those housekeeping genes.
Right.
And so you can just park those to one side because they're just there, that every organism needs. What we're usually interested in are things like bile processing genes. How many organisms have the ability to take a molecule from the host, modify it into something that's maybe less potent or more potent?
Okay.
Things that can interact with drugs.
Right.
So those are, these, maybe you could call them accessory genes. They're not essential to the organism.
Yep.
But they're interesting in terms of the biology of how it interacts with the host.
And that drug metabolizing element, how has that changed in recent years? Like, we're starting to appreciate that these bugs could be impacting on the effectiveness, safety, tolerability of our medicines that are prescribed everywhere.
I think slowly and surely that is a big revolution in pharmaceutical areas. And I think the big pharma companies are starting to wake up to the notion that there's this virtual organ that influences their share price.
Right.
In as much that, it could affect how many people get ill. So nobody wants bad press, but suddenly you're thinking, well, it could be the microbes that are causing these side effects.
Yeah.
B, response to your drug. It could be the microbes that are dictating, A, if your pro-drug's turned into a drug and it's just active, if your drug is just degraded and it's not enough of it's being given, and then if it actually influences the immune system to respond to your drug.
I mean, imagine if, and I don't know how we would quantify this, but imagine if a percentage of phase 3 failures were because the microbiome was doing something to the drug in a population that wasn't really sampled in phase 2 or phase 1 healthy volunteers, phase 2 slightly less sick, phase 3 really sick. They might have a particular microbiome profile. Imagine.
Yeah.
That's huge.
I'd almost argue that you need a phase 0, which is where you do your microbe-drug interaction analysis. Okay. You've gone through your animals, to show, there's safety here. But then you do a really good panel of common bacteria in the gut and ask the question, how does my drug interact with the gut microbiota?
And you do that on the benchtop?
You could do it on the benchtop. You could do it in mice. You could do it in pigs.
Yeah.
There's good animal models for looking at drug-bug, as we call it, bacteria interaction.
I mean, just to push back a little bit, would the best test, though, not be a patient with a disease or a form of the disease, a precursor of the disease, and very rigorous microbiome analysis in the early phase studies? Or would you want to sort of de-risk it even further?
I'd want to de-risk it even further. The problem is, what we know is, A, everybody has a very different gut microbiota. So there's no one person who's a good representative of everybody's guts.
Really?
You might need to do 100 people, which is much larger than the phase 1. Just to take all those different, try to capture all those different gut microbiota that you can have. Because if you look, and that's only just, the problem is that's based on looking at stool. We don't know what happens in the small intestine. So where the drug-bug interaction happens, and depending on what type of drug, let's say it's an oral drug, the first time it's going to really see the gut bacteria in any anger is in the small intestine.
Right.
Unless it's supposed to be absorbed in the stomach, in which case it'll see maybe some stomach bacteria. Right. But let's say it's gastric coated, it gets down to the small intestine, it's released in the small intestine, that's how it's designed. Yep. It's going to see very different bacteria there than it would in the colon, doing very different things metabolically. Those are organisms that are very fast growing, they're very good at using sugars, but they have a wide range of metabolic capacity to actually interact with drugs.
On the microbiome similarity piece, I totally get how if you had 100 random people, 100's your sample size, and they're all random, that there'd be big differences between them all on average. But is there not some sort of, you're sort of almost phenotypically characterising these patients because they've all got a disease. So is there not more similarity amongst them, or do they still have that unique microbiome?
They still have a very unique microbiome.
Right.
I mean, it depends how much they've hammered their microbiome prior to coming to a doctor and start getting a diagnosis. This is always a problem with case control studies where we say, for example, inflammatory bowel disease, the person comes in and they've gone through maybe a whole host of self-medication before they get that horrible diagnosis. Right. You have Crohn's disease, you have ulcerative. The diagnosis nobody wants because that's a life-changing diagnosis. 100%. But they've changed their diet, they've gone online, they've tried prebiotics, probiotics, and also the question is when we see these individuals, how much of the gut microbiota is in response to inflammation in the gut or something that's happening in the gut. And so what we're measuring is a response of the gut microbiota to an inflamed gut, which we know is going to be different to somebody who hasn't inflamed. And so it's very difficult then to use people who are ill as a model a lot of the time.
Understood. Do you think that in the context of these diseases, and this is a big question, is it the passenger? So, like, you know, is it just happening as a result of some other physiological process, like inflammation? Or is it the driver? Like, is it driving the inflammation? Is it driving the disease? Like, what's your thoughts on that? And also, what work are you doing in your current lab to try and kind of tease out these interactions and answer those big questions?
Well, we did have many years ago, I think I'm trying to think 2011, sorry, we did look into the cancer, colorectal cancer question, and try to explore this passenger versus driver concept. Because at the time, the first hit, as it was called, that turns a benign cell in the gut into a tumour, a cancer cell. You know, and the question is, $1 million question is, what is it? You know, because if you can understand that, maybe you can control that pathway of cancer, benign cell turning into a cancer cell, tumour, and all the problems that are associated with that. And there were lots of different things. Diet was one of them, you know, smoking, alcohol, whatever it is. And of course, bacteria. Bacteria is one idea, because there were good examples in the plant world of bacteria causing cancer. So there's an organism called Agrobacterium tumefaciens, which causes galls and growths on plants.
Really?
Yeah, and so for all intents and purposes, it's a cancer.
Oh, wow.
And so what it does, it's got this small piece of DNA, largest piece of DNA, which it inserts into the plant cell and causes uncontrolled growth.
Wow.
And then it allows it to grow in there.
What does it look like on the plant? It's just like abnormal mass?
Yeah, it's just no. It is like a tumour. It's just an uncontrolled growth on the plant. You know,
wow.
It's no. You might have a nice stem. There's a big lump coming out of it, which is not that much different to what you'd see in the colon. You know, nice smooth one, and suddenly this lump, which is going through anyway. So we asked the question, driver, passenger. And we explored on tumour, off tumour bacterial communities and found that the tumour had a very different community of organisms to 5-10 centimetres away. And this was consistent in whichever tumour we saw. And as the tumour developed...
Were you scraping bits off that tumour, or was it excised? And
it was taking a biopsy of the tumour and extracting the total DNA from it and then just interrogating and looking for the bacterial DNA. Yeah,
so the workflow for the patients was maybe some symptoms, maybe... picking up some tiny specks of blood on a stool-based test that's really sensitive. They went in to see the doctor. The camera went down. The camera saw the tumour, take a bit off, and then your lab analysed the heck out of it for what right, okay.
Yeah, and so we did that looking at the bacteria on it, looking at the metabolites, you know, for biomarkers. Yep. And as you can imagine, we didn't get many early stage cancers because a lot of people don't come with it. Unfortunately, people come with later stage. Right, so we did get what's called T1, you know, the early stage cancer. Right. We got polyps because we were looking at people who had a history of polyps in their family.
They're like a precancerous change.
Exactly, precancerous. So, and we had some healthies. So individuals, a lot who were coming in for just because they had blood in their stool. And so they were coming in. And as we said, we always encourage you get blood in your stool, go and see a doctor straight away. Don't put it off. And they were healthy, so they might have had hemorrhoids or piles, and that was the bleeding. But we could get a healthy sample, so we get a small group of healthy individuals, and then we got people with polyps, pre-cancer, and then we get the different stages from T1, where it's turned into an adenoma and it's a cancer, and then all the way to T4. Right. And we get more T4s than we did T1s because a lot of people turn up later in life, and you know it's a bit late then for a lot of them. They will some cancer.
There's options.
There are options. Yes. Yeah, a lot of surgical interventions, etc. But what we could show is that, you know, as the tumours do develop, as they get larger and more aggressive and more, they've died more necrotic. You know, the material inside and they're leaking more. The community changes. And so what we were looking at to start off is we identified groups of bacteria which we thought were the drivers. These were organisms that we think were interacting with the host and driving mutations and driving inflammatory environment. Then as the tumour developed, they all fell off because the local environment changes. Okay. And then new bacteria which are adapted. Wow. And a lot of these bacteria were oral bacteria.
In the colon?
Yeah.
How does that happen?
So we swallow millions of bacteria every day. They pass through, but a lot of the times they don't have a niche or a habitat which they can colonize in the large intestine, and so they get washed. They just go straight out the other end.
Right.
If you give them a tumor, some of them, for whatever reason, they like growing on it. Maybe because
Wow.
they leak amino acids. We don't know. We don't know the full reason, but a lot of the organisms, from teeth and plaque, we can find on tumors.
What about other diseases? Do oral microbes sort of appear? And or is it mainly just the cancer?
I don't think I've seen it in other. We haven't seen inflammatory bowel disease. It seems to be in cancer and on the tumours, and so then we thought of those as passengers. So that's the only example we've really looked. We haven't looked at inflammatory bowel disease. We've just looked at it in the cancer environment. Whether we think that early days there are drivers of inflammation, there are drivers of damage, potential damage, and then as the cancer develops, these organisms can't cope anymore, they can't compete anymore, they fall off, and these other organisms come in.
I also have read recent research that says that the bugs literally move intracellularly.
Yeah.
They can find their way into the actual tumour and propagate its growth through mechanisms, I think, still to be determined, which is just incredible, isn't it?
But it's what microbes do.
Incredible.
They adapt, so any situation, every organism that we will study and can study. Okay, I should step back and say not every organism. A lot of the organisms that we think are health promoting, given an opportunity, they will be a pathogen.
Right.
They're all one step away from being. And it's not like they're waiting, they're sitting there. It's just the opportunity arises.
I see what you're saying.
And if there's the numbers there and the situation right, they will grow in the wrong place. Whether it's, I think we mentioned before. Even probiotics, these health promoting organisms. There are case studies of them causing endocarditis. People drinking lots of them.
Right.
Okay.
And they find the bug.
They find the bug.
And the valves of the heart.
Yeah.
Wow.
Yeah, and that'll be a lactobacillus. Yeah. But those are, I'm going to put people off using probiotics, but those are rare cases.
I mean, on the probiotics, and should we just talk about probiotics a little bit?
Let's talk about probiotics.
This is one of the topics that everyone loves to talk about. So I'll just ask a really open question. Like, what are your thoughts on probiotics?
I think probiotics have a future if they're treated like a drug. I think the thing that's happened with probiotics is many of my clinical colleagues have treated probiotics and just called them probiotics. And so if they've done a study, they've thought, right, I've heard probiotics are health promoting. Okay?
Because by definition, they should be.
By definition, they should be.
WHO definition.
Live organisms, when given in sufficient quantity, should promote health. I'm paraphrasing Colin Hill's paper.
Indeed.
And so everyone thinks, okay, probiotics, they promote health. Now, the interesting thing is, depending on which country you're in, different probiotics promote health. So in the UK, we're very much lactobacillus and bifidobacterium. Go to Germany, you can get E. coli as a probiotic.
Is that the E. coli Nissel?
1917.
That's the one, yeah.
Why
is that?
Because it's cultural. And this is the thing is, different probiotics are cultural. So you go to Japan and they've got Yakult. The Lactobacillus casei. Yeah. And so, that's still the Lactobacillus, but you see that different organisms, enterococci, yeast, Saccharomyces boulardii, that's a yeast, that'll be in some countries. So, again, straight away, we suddenly say, okay, well, if I'm setting up an experiment where I'm using a probiotic in one country, it could be very, very different to a probiotic in another country. And so what tended to happen was, say, a clinician for good intent and good purposes would say, I want to study a probiotic in the context of my disease. Yep. So they would just go and grab a probiotic, a random one maybe off the shelf, or they've seen you could buy, and I'm going to give it to my patients in a randomized clinical trial. I'll do all the right things as if it was a drug. Yep. But the problem is I've just taken a generic probiotic.
Right. They've not selected one based on a particular phenotypic property.
Exactly. You wouldn't do that for a drug. You wouldn't go to your ethics committee and say, right, I'm going to treat inflammatory bowel disease. What are you going to use? Drug. Yeah, but which one? Drug. I'm just going to use drug. I'm going to take the random drug off the shelf over there. Right. And I'm going to give it to my patients. But hold on a minute. You've just chosen, I don't know, a chemotherapeutic. Yep. I just chose drug. Yeah, but that's not going to help your patient. They're going to get very ill on that. I don't care. And so what happened was lots of trials with probiotics turned out to be negative. So probiotics got a bad press. Right. Now, if they had come, if they had gone to the ethics committee and said, right, I'm taking Lactobacillus reuteri because I have this cell data that shows this dampens down IL-1 beta, TNF alpha. It works in a mouse model, and I've got a colitis mouse model, and it dampens down that. So I've got very good evidence that it's specific, and it's not just saying lactobacillus reuteri. You need the strain.
So it's a mechanistic workup to say this is why this is going to work.
This is the reason. This is the rationale for choosing. Not just saying I'm choosing probiotics and I'm going to just randomly take one. So going forward, I think probiotics have a place if they're treated like a drug.
Okay.
You need evidence that it works, building up to a phase 1.
Yep.
You must don't need the phase 1 because a lot of them are generally regarded as safe anyway.
Yep.
Okay, unless you can give me huge doses, and then that's another story. But then you might be able to go into a phase 2A or something like that and say, well, look, we'll take 50-50. We're going to treat these patients over time. Here are my primary outcome, my secondary outcome, and we can see if it works.
So a couple of questions for you. Single strains. Do you think they're, based on what you talked about earlier, some bugs need other bugs to sort of come to life, if you like, or to grow on a culture plate? Talking about hundreds of different species in the microbiome in every patient. So that's an ecosystem. Is a single strain really going to have a demonstrable effect when it's such a complicated system it's going into?
If you think about the site of action, the problem that we've got is our perception of the human microbiome is driven by stool in the gut.
Right.
And a lot of these lactobacilli don't get to the large intestine in appreciable numbers, but they get to the small intestine in appreciable numbers. Okay. So you could load up the small intestine with 50% of these lactobacilli. So, and if you think of the small intestine as maybe being more immunologically active,
right?
It's sensing more compared to the large intestine is quite boring, really. Look, better than I do that the large intestine, some of its roles are to pull water back out. It doesn't have many Peyer's patches, if any. It doesn't do much when I say about, for sampling and seeing what's happening. A lot of fermentation goes on in there, and a lot of important chemicals are made there. But in terms of immunology, the small intestine is where it's at. So if your bacterium is a lactobacilli and you're taking 100 million of them, 200 or a billion of them, then that's going to have an impact in the small intestine.
Right.
So, you could bulk up that small intestine by 30-40%. It's going to be this organism. It'll grow couple of times, and then it'll die out and get washed out. So it could have an impact in the small intestine, and that's where we're very poor at understanding how these organisms interact with the host in the small intestine.
And we're poor at understanding because it's so hard to study, right? It's like, because how do we study it? What do we need to do?
So early days, we studied patients who had ileostomies, so holes in the side.
Right.
And
so it's coming out into a bag. And
you could sample that. So it was going into the large intestine, but then they're a unique cohort of patients, and the question is this small intestine representative of healthy individuals?
And I guess the question for you or the field would be, they've got an ileostomy, okay, so maybe they don't have a large colon because it's been removed.
Yeah.
Does that impact on the small intestine microbiome?
Exactly, and there's a whole load of things. So those were the early forays into it, because you could get small intestinal samples. Then groups like Willem de Vos's in Holland got ethics to do healthy individuals and put a scope down the nose and into the small intestine and start sampling there. But they were very small groups, but it gave us an insight that the small intestine was very different.
Right.
And so to get to the small intestine, you can't sort of ask 100 people, would you be willing to be scoped? And it's not pleasant. I don't know if you've had it or not.
I haven't, no.
I've had small scopes put down.
I've watched it.
Yeah, it's
of course, I think it just feels really unnatural, because it is. How often would we have over time had big tubes stuck down? No,
and there's risk associated with something like that.
There is.
And if you want to do a longitudinal study where the patient's coming back 10 times, to have that tube put down 10 times, it's not pleasant. So it becomes very difficult. But that being said, there are companies who are producing engineering solutions. So there's a company in Canada that we work with called Nimble Science, who have a little capsule that you can swallow.
Oh, okay.
And it opens up at different parts. You can follow it.
So it's like a little robot?
Yeah, so it just opens...
Scoop. Back in.
Opens up and it samples maybe 100 microliters, shuts up, preserves it, and then when you poop it out, hopefully you catch it and you don't flush it away, but you poop it out.
Of course, you need to recover it. Interesting.
You need to recover
Very
it.
interesting. I was just about to say that, where does it go? But of course it goes there. Okay.
As long as you've got no strictures or anything in your gut to allow it. I think they might give you a proxy beforehand to show
that would make sense.
Yeah, and I think a couple of them do that. But
so we're innovating, and but generally speaking, what are the high level differences then between the small bowel and the big bowel in terms of microbes? Like, what have we found?
So the small bowel is much more dynamic for obvious reasons, because it gets fed really 3 times a day with a high energy diet.
Yeah.
Which the large intestine doesn't. The large intestine gets a trickle of poor energy source material. In other words, lots of carbohydrates that you can't utilize, whereas the small intestine gets lots of sugars, lots of fats, lots of proteins. And so the organisms there are much more adapted to grab sugars and grow quickly. And so what you get are things like lactobacilli, enterococci, E. coli, streptococci, Viennella, these organisms that are very good at metabolizing sugar and growing quickly. But it's also lower density because it moves quickly. So these organisms are fast growing because they need to grow quickly to maintain their presence in the gut.
Makes sense.
But then things move through quite quickly. The transit through the small intestine is much faster than the large intestine. And so it doesn't lend itself to produce dense, complex communities. So there are fewer organisms there, it's not as dense, and there's a lower abundance of them.
Got it. Okay. And what about functional properties? Have we done much on that, or are they very similar?
No, they are. No, the fermentation, does happen in the small intestine. You do get these short chain fatty acids being produced, but I don't think that. I think we haven't had much luck in really analysing it in full detail because it's difficult to get to.
Got it. Let's make a mental note to talk about this in the context of FMT IMT later, because I think it's super interesting.
Yeah.
Now back to probiotics. I'm sure as a professor and an expert in the microbiome, people ask you all the time, Julian, which probiotic should I take?
Yeah.
How much should I take it? Should I try this? Should I even take probiotics? I get it too.
Yeah.
And I'm very often saying the kind of same thing, which is, look, I don't take them for these reasons, and I would spend my money on the best food I can possibly afford.
Yeah.
What do you say?
I say exactly the same thing. I
Do
think
you? That's good. That's reassuring.
Well, no, it also comes down to, it's the same thing with vitamins. The more and more we see is a majority of people who don't take vitamins aren't walking around with rickets, scurvy, beriberi, all the deficiencies that you used to see.
Yeah.
Because our food is reinforced with it. We're getting enough vitamins, okay? Yeah. Probiotics, again, for the healthy individual, this must be no need for it. Yeah. Where they do come into their own is if you say you've had a course of antibiotics.
Yeah.
Okay, and you might have had a bit of diarrhea from it, then there's no harm in taking probiotics there. Which ones? It's a good question, because a lot of probiotics don't contain what they say on the label. Some companies are better than others.
Okay.
I'm not on commission with anybody, but for example, Yakult. I've opened a bottle of Yakult, and we have grown the bacteria from there, and it is what it says is on there. Okay,
that's good.
Danone, the same. If they say those little Danone says it's got X amount of lactobacillus casei in it, generally it is that organism. There's nothing else in there. We work with a company called Cultec, who make something called Lab P4 and Lab 4 for boots. I've opened that up, and it has got in it what it is. So there are some companies that do.
But how do you select? Because what you're talking about is you've got lacto, Saccharomyces, E. coli, Nissel, some have a blend. What should you do? And I think the antibiotic case is good. In Italy, they give probiotics.
VSL3 and things like that, which is a mix of probiotics.
They do, Ferring VSL3. I mean, that's the one that I would typically recommend because there's a lot in it.
There is a lot in it. And that way you're hedging your bets.
Right.
And that's the problem is, just taking one, it's, well, what is it doing and is it fit for what you want it to do?
Right.
So without testing it, I would hedge my bets. VSL3 or the Lab 4P, which has got 4 organisms in it. You're covering your bases sort of there. Yeah. And it's a difficult one. It is. I don't think we're at the stage where we're going to be getting prescribed probiotics by the GP for something at the moment, because the evidence isn't strong enough at the moment.
In the absence of data. So thought experiment. If you had every option available to you, including, for example, banking your own microbiome before you got antibiotics, or taking a sample that had been derived from a super healthy person, or kind of any specific strain or consortia, what would you ideally want to have? Where should the industry and the researchers be shooting for post-antibiotic use?
I like the banking your own.
Yeah.
Because it's interesting. You mentioned that super healthy donor.
Yes.
So we've got projects where we're looking at liver disease.
Yes.
And maybe Ben will talk about it later, And we're talking about transplanting microbiota from healthy individuals into people who have liver disease, fatty liver disease, and seeing if we can change their chemical and metabolic landscape. Now, when we decided who should we be choosing as a healthy donor, I argued we should be choosing somebody like myself in their mid-50s, in their 60s, okay, who have not abstained from alcohol over life. I wouldn't say abused it, but not abstained, Okay, not clinically obese, and has good liver function.
Yep. I see where you're going with this.
And a good FibroScan score, say. So my liver's not stiff, it's about 4.5, so it's nice and wobbly, my liver, it's not too stiff. But what that tells me is maybe I have a gut microbiota that
is protective.
However, if you take a 20-year-old who can do a triathlon in 2 hours or less, okay, when you pick them up at 60, you might find that actually they've got fatty liver disease, they've gone obese, and actually their microbiota was not protected. Just because they look hale and healthy now, and this is the one thing, we can never predict what is a healthy microbiota because it's all context dependent. So if you could fast forward and take a time machine and look at them in 40 years' time and say, no, their liver's still good, so let's say that actually that is a protective microbiota. And that's the sort of one we want to transport. So
interesting, because the guidelines, the guidelines that Ben was first author on, joint first author, I think. Absolutely. One of my, there's a lot in there we can talk about. We could talk about so much in there actually. Like they have they have lots of exclusions that are theoretical. Yeah. Right.
Yes.
But I suppose one of them is there's a cutoff. I think it's 60, maybe 65. There's that ballpark really is what I see typically in FMT protocols and guidelines. And by the way, if you don't know what FMT is, it's the movement of microorganisms from one person into another. And
fecal microbiota transplant.
That's it. And the microbes are derived from stool. And there's various protocols, procedures, systems, checks and balances to make sure that that material, which is the starting material for what goes into a person, is as safe as it can possibly be based on our current understanding. And there's a mixture of pathogen screening through analytical methods, but also a whole variety of questionnaire-based exclusions.
Lifestyle questions.
Yeah, and we're speaking specifically right now about age. And it's like, I have pushed back a little bit and said, well, if someone's 85 and they look incredible and their biochemistry and everything is phenomenal, they've never had antibiotics, veggie diet with 100 grams of fiber every day, and they're running marathons. Okay, that's sounding like a pretty good donor, right? But they'd be cut out. Yeah. Whereas your 25-year-old who's just come back from Ayia Napa,
I know, you
know what I'm talking about?
Just been living off Red Bull.
That Red Bull and vodka and chips. Maybe some promiscuous behaviour. Likes to go out with the lads. Maybe had a couple cigarettes over there, but nothing too much. It's like, okay. So they would qualify, but somebody else wouldn't. So that's why I think there's a huge amount of art and knowledge and know-how in donor selection.
I agree with you totally.
So important for this.
I think this falling down. It's an easy trap to fall into, saying somebody's young, they must be healthy. No, what's their life trajectory?
Yes.
Where are they going to go?
Yes.
For all you know, they could hit 40, 50 and have colorectal cancer.
Yeah.
So what you've just done is transplanted, potentially, if we buy into it, a microbiota that could be carcinogenic or pro-cancer supporting.
Indeed, but there's checks and balances there as well. There's the fit testing for blood in the stool, and then there's a first-degree relative exclusion on if you have a first-degree relative with colorectal cancer, I'm afraid you can't be a donor. So
But let's say you take those out, unless you're going to follow that individual.
Sure.
We ask about mental health, psychiatric problems.
Yeah.
But but going back to your point about, and I think it is a valid point, that a lot of the things that we do speculate on being a gut microbiota driving it, at the moment the evidence is very mouse driven.
Very weak.
It's weak. And so, are we excluding a lot of good donors based on what we thought is, oh my gosh, this may happen, this may happen, this may happen, saying, well, where are the bodies? It's that sort of thing is, if you're going to exclude, you really need strong evidence. Exclude on pathogens, they're clear.
For sure. And being as robust and rigorous as we possibly can on pathogens, I think is totally fundamental.
Well, as we know
Indeed.
from the, was it 2019?
2018-19, the Philippe paper.
Yeah,
yes,
the two individuals who were given.
Hopefully, I've done their name the right kind of pronunciation there. But indeed, two individuals.
Immunocompromised.
E. coli bacteremia.
Yeah, one died.
One died. Yeah.
And but interestingly, that was because they hadn't followed the procedures.
Indeed.
And and I think Ben and Nabil's protocol then would have caught that, because they did have ESB, it did have antibiotic resistant bacteria, E. coli as a so.
I mean, and we're going even further at Intrabiotics in terms of the sensitivity of the assays, the frequency of the testing, and just the overall kind of hierarchy of checks, balances and controls to reduce risk and increase the assurance at each stage. And ultimately, I think that if we're going to be doing this type of approach at scale, and I really mean at scale, I'm not talking 10 people, I'm talking like hundreds of thousands of millions of people, we must have those checks and balances in place, and we must be absolutely robust and rigorous on all elements associated with starting material safety. Now, going back a step, what did you do for this liver study? Who won the debate? So are people like yourself who've had a couple of drinks or so and so on? I suppose we can't just have abstinent donors. Is that really?
I think that's impractical. I mean, at what we fell back on the we fell back to using metabolic profiling to decide on our donors.
Oh, you're doing that?
Yes.
This is interesting. I've only seen microbial driven selection.
Yeah.
In liver populations, the short chain fatty acid producers, essentially, right? Yeah. Fascinating.
So this is looking at
really
these are the healthy individuals metabolically. They sit in this area of the graph. Here are fatty liver disease. Probe Ben for this later. He'll have much more information.
I will do. I'll make a note to do. We should also, while we're here, talk about the metabolomic toolkit, because we spoke about the metagenomics.
Yes. Yeah.
And maybe before we jump into this, what is the metabolomic toolkit? What is metabolomics? And
so, as I was mentioning before, we're very good at DNA-based approaches. I think, we've got to the stage now that everything we do with DNA-based approaches is incremental. There's nothing sort of revolutionary coming down the line. We're tweaking this, changing that.
Right.
But what we've realised, or my group has realised, and we're studying, is that in a healthy individual, the communication between your microbiota, the organisms that live in and on you, is predominantly via the metabolites and the proteins they make. So whilst we're not very good at looking at the proteins, and that's more technical and it's more information-based problem that we should be able to solve later down the line, we're pretty good at extracting the chemicals they make, the metabolites, analysing those and determining then, because we can buy them, what they do. We can incubate them with some cells, we can put them into an animal, we can put them into animals then with genes knocked out and see, well, if that receptor's missing, does the animal respond to this chemical? And that's how, we find out things like short-chain fatty acids have their own receptors, and those receptors then are found all across the body, and they're fundamental to mammalian biology.
Right.
And so these metabolite profiling methods called metabolomics and metabomics are the tools that we use, and they're very powerful methods that we can apply to longitudinal studies over time, case control studies, so two groups, with and without disease or with and without an intervention. And we can take plasma, we can take urine, we can take fecal water, which is feces bulked up with water and just filtered. We can take swabs, and we can just look at the metabolites in there, and we can look at lipids, we can look at small molecules, we can look at a whole range of complex molecules, and so that's what we really do at the lab at Imperial, and then we sort of associate that with the bacteria there and try to make the connections between them.
So you think that the things that they produce might be more important than just their presence, right?
Yeah, I think it is, yeah. And the interesting thing is you don't need much of some organisms, so they could be very low level of organisms. You don't have to have it, it doesn't have to be 90%.
Fascinating.
It can be 0.0001% in your gut.
Fascinating.
But it can have a significant impact, making metabolites that accumulate, and then just switching on genes left, right and centre.
So what are the kind of big buckets of metabolites that bugs produce that we think might be important?
So the one you've mentioned already, the short-chain fatty acids. We now think there are 4 of them that were important. It used to be the trinity: acetate, propionate, butyrate. We've added to valerate to that.
And they're named based on how many carbons are...
Yeah. So acetate 2 carbons,
Yeah.
propionate 3, butyrate 4, and valerate 5. And they've got proper chemical names that give you a clue. Like valerate is the generic, but it's called pentanoic acid, and penta is 5.
So we started with 3. Then it became 4, and now your group has done some work to say actually maybe it's 5, or it could be more.
We started with 3.
Yep.
Acetate, propionate, and butyrate. That's the trinity that you see a lot of write-up. But we think the valerate's now important.
Yeah, and you've done some very elegant work in what I'll call reverse engineering, or I guess another phrase could be elucidating the mechanism of action for an FMT in C. diff.
Yep, which was in the news yesterday.
I did not see that.
Yep. Nice have said that.
Oh, yes.
The BBC covered it, Guardian picked up on it.
Yes.
And actually, they did mention about superbugs and antibiotic resistance, which was interesting.
No, I know Nice released updated guidance with a recommendation for the use of FMT in recurrent C. diff. I didn't realise they mentioned anything about valerate, did they?
No, they didn't. Okay, no, but it was. It's fact that it just hit. It got into the headlines. I was surprised as well. But it got
all over the BBC, all over the Metro, everything.
Yeah,
fantastic.
A lot of people picked it. Well, a lot of news outlets picked up.
Fantastic.
Yeah. So that was great. Yeah.
Really, really good.
And so we've used this FMT as a tool to understand the role of the microbiota in the host, because it's one of the only times that we're allowed to totally transplant somebody's microbiota and change it. It's difficult to take a group of individuals and say, we'll give them antibiotics for no reason. But this is an opportunity. So people who are sick, we can actually just modify the gut microbiota quite dramatically by putting somebody else's microbiota in there.
I mean, it probably is the most dramatic.
It is,
has to be, right?
Well, apart from bariatric surgery.
Oh, okay.
Which is super dramatic. That's the other really interesting experiment that we're allowed to do. Well, say allowed to do. If people are undergoing gastric bypass.
Oh, wow.
Especially where they bypass the stomach, the Roux-en-Y gastric bypass, which is the gold standard.
Yeah.
That, oh my gosh, that changes your large intestine. Into a small intestine, basically. Microbiologically. It really, really, really alters it. So that's the other one. So there are a couple.
Fascinating. Well, hold on a second. So this bariatric surgery is phenomenally good at reducing people's body weight.
Yeah.
So there's definitely a sort of physical capacity piece, isn't there? Where you
yeah.
But
you bypass the stomach, so you're not taking as much food in.
Yes.
The stomach's still there.
Okay.
In situ, it's still there.
Yeah.
But it's not plumbed into your small intestine.
So do you know there's a company that, and I don't know if it's still going, and I'm sorry to the company if they're doing really well, they were giving capsules containing oxygen to people. I look at your face.
I'm just trying to work this out. Okay.
I hope I'm not...
an empty capsule.
I hope I'm not doing them a disservice, okay? But hear me out. So what they were saying, or their hypothesis, and again, apologies to the company from doing you a disservice, was you do this bariatric surgery and there's more aerobes which appear in your gut where they weren't before. And their hypothesis was that it was the aerobe change, the shift in whatever it was that was driving suppression of hunger and so on, so on, so on, so on. So I think they were literally giving a capsule that, okay, we shall say no more on the matter, but I just thought it'd be very interesting. So how much of this effectiveness against, obesity is driven by the physical bit, or is there a microbial bit as well?
There's definitely, obviously, the physical bit. The calorie restriction is no brainer. Right. People keep saying obesity, how do you do it? Well, it is thermodynamics.
It
is simple thermodynamics.
It's not that.
People don't make fat from nothing. Okay.
Yes.
If you take a mammal and stop feeding it for a day, it starts losing weight. I know, and I'm not trying to trivialize obesity.
I hear you.
But at the end of the day, it is energy in, energy out.
Yeah.
It is a simple thermodynamics. So
the question for you...
Stopping the energy in is difficult.
That's exactly what I was about to ask you, so I'm really glad we've arrived there.
One of the things we don't seem to be... We don't want to do is hold the food companies' feet to the fire and say, actually, this is big problem that you've caused by the foods that you produce that are obesogenic, that are cheap,
low fiber,
micronutrient
light, all that stuff.
Thing, ultra processed foods that taste great, cheap, and actually are driving this. So that aside, okay, and I'm not going to point to any companies. They're all as guilty as each other.
We do need to talk. Sorry, we need to make a note. I would love to pick your brains on the communication between the gut and the brain, and are the bugs adapting to these foods and going, hey, we want some more of that? And
that's a good question.
You know,
I don't know enough about you want to get somebody like Kevin Whelan or Gary Frost in.
Yeah.
Kevin's
on my shortlist. He's a very interesting guy.
He's good because he knows about his emulsifiers. He knows about all the stories there about that. So he'd be better. I don't know if the gut microbes are adapting to them. Maybe small intestine. I don't know. I wouldn't put any money on it at the moment.
Because some people some people think that cravings for chocolate and sugar, there's some sort of neuronal cellular circuitry stuff that's going on here. However, it might be that the bugs go, where's that gone?
Yeah.
I want some of that.
Yeah.
And they produce, because my understanding is that on the metabolomic side, we haven't characterized anywhere near all of the products of microbial metabolism. No. So there's a lot of unknowns.
There's a lot of unknowns.
So they might be sending signals, molecules they produce that trigger some sort of craving in the brain for sugar.
Yeah.
It kind of makes sense, right?
Yeah.
Now, going back a step on the short chain fatty acids. So is it the case that there's some sort of kind of co-evolution thing here where we eat something that's rich in fiber and our digestive enzymes in the upper part of the GI tract can't break it down. And then it arrives in there as a food source and they go, yummy. They eat it and then they produce something that benefits us. Is that what we're seeing?
Definitely. I mean, if you think of the range of functions the short-chain fatty acids do, they're anti-inflammatory, anti-proliferative, so they cut down your risk of getting colorectal cancer. They may help with your mental health. All the sort of things that may allow you to last a little bit longer, reproduce and pass on the genes to the next generation and the microbiota. These microbiota will be maternally transmitted. So if it's a group of organisms...
Say that again, sorry, they're maternally transmitted, so they're coming from mum?
Predominantly. Yeah. If we're going back hundreds of thousands of years, the only place they've been coming from is from mum in natural birth.
Yeah, okay. The early life microbiome you're talking about, because there's no C-sections.
No C-sections. Yep. So those microbiota that were interacting with the diet, benefiting the host in terms of health, allowing them to have more children, reproduce, that microbiota then becomes like a genetic information that is transmitted to the next generation. Right. And so of course I see an interaction. It's a win-win for both. The microbiota has a nice home to live in. It gets fed things it likes to eat, and at the same time produces metabolites that the host responds to, whether it's bile acids that help with driving, minimizing obesity, or gathering energy from food as it comes in, whatever it is, down to neurotransmitters that make the host feel happy, to short-chain fatty acids that are anti-inflammatory and also protective against foodborne organisms coming in and killing the host. All those come together, without all the other parts that we don't know about. There's those simple players there. They all play a role in host health.
Amazing. So there's this element of, we kind of know what we need to do to be healthier in terms of eating, dietary habits, so on. But what's actually happening is we're feeding this orchestra. Yeah. And it just starts playing this incredible symphony. And all the things that it's producing in its theatre of activity, or not all of them, but many of them are actually benefiting us. Yeah. So we're now starting to define why these foods actually make us better and reduce the incidence of disease and so on and so on. And it's what you're saying is metabolomics that's giving us that insight, yeah? At
the moment, that's where our effort and concentration is, but the proteins will come to the fore. We'll understand some of the proteins they're making. Yeah. And the reason we're not really at that point is because, as I said, it's just technological. The proteins...
What happened? They degrade fast?
No, it's more to do with identifying them. Okay. This goes back to the fact that identifying a protein relies on very well annotated bacterial genomes. And if we're not annotating those very well, because we either don't have the full genome, we can't come back. Our protein just comes back as an unknown again. Just like I said before, 60 to 70% of genes coming from a gut are unknown. Right. It's the same sort of thing with proteins. We just see a protein and say, well, there's nothing in the database that tells us what that protein does. Right.
Right.
You know, I'm not saying it's super better with metabolites. We still, as you mentioned, you know, a lot of metabolites, we see them, the structures, and we think, oh, I have no idea what that is.
Wow. So that could be a cure for something.
Yeah.
There's a paper published in Nature a couple of days ago. They were mining the microbiome to find antimicrobial solutions to, I think, Pseudomonas.
Yeah.
I don't know if it was agenerosa, the one that predominates in cystic fibrosis.
Yeah.
I can't remember. But my take home was, right, the cure might be residing inside of us.
Yeah.
The cure to all these things might be in that, whatever it is, 2 million genes that sits within all of our gut. I mean, that is incredible.
That is interesting about microbes is, I think, Craig Venter, the guy who was involved in the human genome.
Incredible entrepreneur, right?
Yeah. And many years ago, he says, I'm stopped looking at mammalian genomes. They're boring. Because every time I sequence them, I don't find anything new. Whereas bacterial genomes, every environment we go to, we keep adding to the list of new genes. Incredible. It hasn't plateaued at the moment.
Incredible.
It's you know, some people think it's almost like an infinite resource of genes in the microbiome.
Incredible.
Because when I say the microbiome, I'm talking about the Earth's microbiome. Because, you know, a lot of people forget that below the sea floor,
there's a microbiome.
It's the majority of biomass. You know, 10% of the world's biomass is below the sea floor, and it's bacterial. That's a lot of biomass. Yeah, it's a lot of biomass.
Wow. And you've got these bugs which are extremophilic, so they can live in these vents that are like 135 degrees Celsius. And that level of adaptation, you know, it just exemplifies how phenomenal these microbes actually are.
I was in House of Parliament just the day before Boris Johnson resigned. Okay. It's a great story, this, because we were going to go into our room. Right. For an all party
the special thingamajig on and there's definitely a more technical term than thingamajig, isn't there?
Yeah,
so
all party parliamentary group, APPG.
Yes.
And we couldn't get into our room because Boris had hijacked it for a meeting. So he came out, we went in, and they were asking me, you know, antibiotic resistance.
Yeah.
Will we solve it? And I said, it's solvable. Yeah. Okay, because microbes make so many antibiotics. Yeah. The problem is not finding them. The problem is an economic. That's why Lord O'Neill was brought in to try to find an economic solution to.
And it was a good one. But it's not happened yet.
Not happening. No. But a pharmaceutical company putting in £2 billion to develop a drug that then sold for £5 and then goes out of use after a year because it becomes antibiotic resistance. So, I think you're right. The solution is there.
Could be there.
In the gut, soil, marine, coral, wherever it is.
Yeah. Well, when I become Prime Minister, I'll make sure that we've got proper pool incentives for antibiotics. You know, it's too short-sighted to not be investing massively in novel antimicrobial solutions, because it will hit us, and it will hit us so hard, and everyone's going to say, what were you people doing? Where was the recommendations and the infrastructure? And we said, well, we knew, but we just didn't have the appropriate resources available from the government.
It's a global problem. I remember writing to my MP years ago saying, look, the organisms in India don't care about boundaries. So if antibiotic resistance pops up in that country or in the UK, whatever it is, it's going to move globally.
Yeah, I mean, we're seeing it though.
We've seen it with COVID, how quickly things move globally.
And there are people out there, I understand, through discussions with infectious disease clinicians, there are people out there who have untreatable gonorrhea right now.
Yeah.
They cannot treat it. So it's now lifelong until someone comes up with something really novel to get rid of it. That is scary.
And it's scary because then there's a lot of people using antibiotics prophylactically.
Very scary indeed.
In groups, you know.
Very scary.
And, you know, they buy it illegally over the web.
I understand.
Self-treating.
And
then you're thinking, gosh, how many STIs are going to start popping up?
Indeed. No, Julian, absolutely. And it's still very much we're taking a very broad brush approach to treating something that we could treat very specifically with precision, for example, phage and so on. So things I'm really keen to talk about with the remainder of the time. More on FMT.
Yeah.
Let's also talk about the other elements of the microbiome. The microbiome.
Yeah, the fungi.
The fungi.
The viruses.
The viruses, yeah.
It's a community. So they all come together to form the community.
Yeah.
Now, fungi are interesting because many years ago, we looked into this on the back of the fact that a paper being published that said the mouse gut is full of fungi. So we thought, oh, okay, that's novel. You know, there's a picture of all these different organisms. Okay, so we looked at the human gut and we didn't find many. Well, it was a stool sample, but we found very, very few. So what we did is we tried to grow them. So we tried a dual approach, culture independent, again, DNA based.
Yeah.
targeting fungi and those organisms, and a culture-based approach. Try to grow something from stool. And what we did is, when we did the culture-based approach, we grew your candidas, your Candida albicans, your Saccharomyces cerevisiae, your baker's yeast. And if we did the culture independent, they were very, very low abundance. They were very low. And we found some organisms, one called Blastocystis hominis.
Oh, I know about this.
Now, it's interesting.
Tell me.
Well, it's interesting because Blastocystis hominis, I think, has got a bad press.
Well, as you know, some guidelines for donor screening for FMT say if you've got Blastocystis in there, sorry, but they can't be a donor. And there's also the other one, which is fragilis. I can't remember the name of it. Yes, indeed. Yes.
So
we'll ask Ben when he comes on.
Yeah, Blastocystis hominis, I think, is like E. coli. I think lots of people have it in their gut. Some people have it as a parasite. But
is it Dientamoeba fragilis? No,
it could be it could be an entamoeba.
Yeah, I can't remember. Anyways, we'll get Ben on and we'll interrogate the lead co-author.
You know, and we found, like, in this Irish, because I was in Ireland at the time, this Irish population, that nearly everybody of the 30 or 40 people had Blastocystis hominis, but they were all healthy.
Wow.
And I thought, it's like having E. coli in your gut.
It can be, and
but it's the strain. And the problem was the literature. A lot of people had published Blastocystis hominis papers in parasite journals. So straight away it had this name above it. It's a parasite.
Right.
And yet I think it's a commensal eukaryote, not a fungi, but a eukaryote.
Yeah.
So a complex organism that happens to live in lots of people's guts. And in some people, just like E. coli, if it was 0157, it will cause a problem, diarrhea. But in most people, they just carry it. Doesn't affect them whatsoever.
Yeah. And with regards to donor screening and such, like, I guess this is another example of we're taking perhaps an overly cautious approach because we don't yet truly know, right?
Yeah.
So how important then are the fungal elements and the phage and the content?
The phage are fundamental to the gut microbiota. Okay, the fungi I think are much more opportunistic. Again, hit them with some antibiotics. If you've got HIV and you're immunocompromised, then they have opportunities to grow and cause problems. Basically, most of the time, fungi don't cause a problem.
Yeah.
I hear some people say, I've got a candida overgrowth. So-and-so, my health person, whoever they've gone to see, their health guru told them, what you got is candida. No, it's not, it's candida. And you mostly haven't. You know, this candida albicans thing, I don't buy into it.
Right.
Okay. It'll pop up in certain situations. You know, thrush, we understand that.
Right.
Oral thrush. Like, you know, when I was younger, we were given antibiotics in sugar solutions.
Right.
And it used to stick around the mouth and cause oral thrush because the organism, the bacteria were depleted and an opportunistic fungus would be allowed to grow. But for the majority of people, we rarely see any issues with candida. Now, fungi. Now, that's not to say that there aren't situations where it can become invasive, it could become a problem, but I think those are far and few between. Now, the viruses, every one bacterium in the gut has 10 viruses trying to predate it. So viruses are the most abundant organism on Earth.
Is it really a 1 to 10 ratio?
Yeah.
That's mad.
And I might have got this figure wrong, but there was, if you take every bacterial virus in the world and put them end to end, okay, so make a chain of them end to end, do you know how long they would be?
It's going to be something mad, isn't it?
I think it's 150,000 light years.
No, it can't be that big.
It is. I'll come back
to you. It can't be that big.
It is phenomenal.
Whoa. We'll make sure we'll check that.
Think
but
of these,
that's
they're 10. You know, they're 15 nanometers.
In my head, I was thinking, right, it's not going to go beyond our galaxy.
It spans. It goes way outside our galaxy.
I can't believe that.
It's
that
light
is
years.
So
it's
cool.
Light years. It's either 50,000 light years or it's huge.
That's so cool.
And that just tells you how big a number there are. Incredible.
But
that
is so
do is
cool.
They play a real fundamental role in the dynamics of the gut.
Wow. My mind's, to be honest with you, Julian, you've successfully blown my mind.
So there are lots of viruses in the world, and what they're doing is, as an organism grows, the chances of a virus meeting it increases. So as the population, say, of one organism grows, suddenly the viruses will attack it. they'll kill it off, it'll drop down again, and another organism come up. So if you could follow, say, in real time, populations of bacteria in the gut, what you'll see is them going through phases. They'll grow up, drop, grow up. So they'll go up and down, up and down. And what that's showing is predation by viruses. When they drop down to a certain level, there's not enough viruses, not enough bacteria around, and the viruses wash out, and then slowly they'll grow up again, and they'll get to...
goodness.
And so you see this sort of dynamics of populations going up and down, and a lot of time viruses are driving that. They do it in the oceans. They've been implicated in global warming and climate change, and because those viruses are having impacts on bacteria, which have impacts on oxygen levels, carbon dioxide levels. So that they're fundamental.
So we've got a very bacteria-centric view, don't we? And what are the drivers for that then, in your opinion?
Ease of growing them to start off with. Even, I said, going back, I said it was we carried this baggage from environmental microbiology into the gut, saying we can't grow them, so we're going to use DNA-based tools. Not that long ago, Hilary Brown and Trevor Lawley from Sanger published a paper to show that you actually can grow nearly 90-95% of bacteria in the gut.
So they pioneered...
They actually just put the effort in.
Oh, really?
And it was a case... I remember talking to a guy called Steve Giovanoni years ago, and he was a big environmental microbiologist, and he identified this group of organisms in the Sargasso Sea called I think it's called SAR11 group, and they were very abundant. But he said, I can't get a PhD student to come to the lab to grow it because it's just not sexy. Nobody wants to do good old-fashioned culture.
So you just got to put the work in.
Hillary and Trevor did, and his lab put the work in, and they weren't using any special media. They were using the general media developed by Harry Flint. And it was they just put the effort in and grew things for months at a time and picked off and banked them and then analysed them genomically and found, well, actually, we're growing nearly everything in the gut.
Oh, wow.
And so, but it comes back to that problem is, it's not practical. Trying to culture something for a long time, it's just not practical.
I'm going to try and get Harry on and learn about his journey. He'll be an incredible guest.
He's an elder statesman of the gut microbiome.
I want to get them all on and just hear their story, and I'm sure he'll have so much to say. So we're talking about the viral element then, and so... Where are we going to go, do you think, with the viral piece? Do you think we're going to start seeing more and more studies that say, actually, this is really important? Or do you think because 50% of the stool that comes out of your body are bacteria or is bacteria, then therefore it's worthy of the most inquiry and resource?
I think because they make the metabolites. I mean, what would be interesting with the virus is twofold. A, they could be used maybe as biomarkers for the presence of an organism.
Yep.
Because they are more abundant than the organism.
Yep.
So if you can... I mean, the problem with viruses is they need a host to grow, and you can't grow them in the lab without a host. You can't do what you can do with bacteria. So you need genetic approaches to look at them.
Yep.
So that's one way. They could be biomarkers, sentinels for presence of an organism, change of state in the gut.
Yep.
That's just thinking. Or the other one is they can be used as an alternative to antibiotics. But not every organism... it's easy to find a virus that'll kill it. For example, C. difficile. A lot of the viruses there are what's called lysogenic. So the virus doesn't lyse the organism. It doesn't pop it open. It doesn't get in, reproduce, and then burst out like Alien from the Alien film. It goes in and it integrates into its genome of the host and just sits and does nothing.
Oh, right.
And then maybe later on some environmental trigger. It could be starvation. It could be like it suddenly triggers it, and then it starts reproducing and pops out.
Yeah, got it.
So you can throw it in, and nothing happens. The organism just absorbs the viral population, and nothing happens. So that it will come to the fore. Some diseases, bacterial diseases, there may be a role further down the line for phage therapy, as it's called.
Yeah. And can phages be harmful to human health, or...?
I don't think there's any evidence.
Because they predate on the bugs,
yeah, that's a great thing about viruses. They're very, very specific for their host.
Yeah.
And when I say, okay, we get a bit of transmission, we've seen it with COVID, we think it was an animal to human jump. But I don't think we'd ever see a plant to human jump, of a virus. And a bacteria, it's just the jump is too big.
Right. You don't think it came from a lab in Wuhan?
No. I think the recent WHO report that came out that I was reading. No, I mean, you never say never because you can't, without having samples and looking at it and having all the data, but all the evidence points to the market.
Okay. So going back a step then, on the phage side of things, probably not harmful to humans, very specific for bacteria, big potential in treating antibiotic resistant bacterial infections. What about in the context of IMT, FMT? So you've done a lot of work with Imperial on FMT, IMT. And I mean, I probably shouldn't try and summarize your work for you because you're right here, but you've done a lot of super cool stuff, including, I think, producing the most elegant elucidation of the mechanism of action for FMT in a disease, C. difficile infection, right? So tell me about your work on FMT, IMT, and we should definitely talk about the C. diff thing, because I want to press you a little bit on, can you apply the same tools, techniques, principles to try and unravel the mechanism of action in other diseases, because that could be so impactful. So tell me the FMT, IMT story.
So C. difficile is an infection, bacterial infection of the gut, driven by antibiotics. And what happens is people have an antibiotic, they get this infection, and what we realised early on was the reason that they weren't getting an infection was that the bacteria was suppressing the growth of C. diff. And what the antibiotic was doing was removing those bacteria that were stopping the C. diff from growing and causing providing an opportunity for C. diff to grow. So it was an ecological problem. What we needed to do was get the bacteria back in to put that suppression on the C. diff, and obviously IMT, intestinal microbiota transplant, or FMT, would seem to be the best tool to do that. But it's not without its risks. As you mentioned before, there's the whole donor screening, there's a whole donor retention. Just getting donors is not trivial.
It's hard, right? It's hard.
Yeah, and then we do it by a nasogastric tube, so tube down into the duodenum. You have risks there where you can go in the wrong hole, and you might put it into the lung if you're bad gastroenterologist and you happen to do it badly.
That would be very bad. Please don't do that.
Yeah, don't do that. But the whole, and it takes, you need a gastroenterologist, you need all the different things to go with it. So we decided to follow a group of individuals who are having this and then try to model it in the laboratory. In glass jars, we modeled the colon. And when we say we modelled the colon, we modelled the colonic bacteria. We didn't have cells from the host. We just had the bacteria from a stool sample grown in a jar, and we mimicked what would happen if you did a C. difficile FMT. So we put the organism in, we gave one vessel saline, salty water, we gave one vessel an FMT, and then metabolically we followed over time what happened. And we also looked at enzyme activity and a whole range of metabolites, bile, etc., etc.
So using the metabolomic toolkit that you're talking about.
Exactly, because that's what Imperial are very good at. They've got this National Phenome Centre with all these fancy machines that can look at the metabolites.
Yep.
And what we found is in the vessel that consistently got saline, one compound never changed. It just flatlined.
Interesting.
So it was high to start off with. We put the antibiotic in and C. difficile to mimic what happened in the host. It dropped down to 0. Give it saline, nothing happens. Give it an FMT, it recovers.
Wow.
And so that compound was valerate.
Right.
And then what we found was valerate, when you incubated with C. difficile, stops C. difficile from growing. But it doesn't stop anything else from growing.
Valerate, going back to what we're talking about earlier, is a short-chain fatty acid.
It's a short-chain fatty acid.
Produced by the bugs.
Produced by the bugs, and only produced by the bugs. It's not in your diet, unless you're taking valeric acid from plants for being calm, but very few people who got C. diff are doing that.
Right.
And so we found that that's one aspect of it. But C. difficile has a complex life cycle. It can turn into a spore, and so it can sit around in your gut if it sticks to something, waiting for the right opportunity to germinate and turn into the cell, which then produces a toxin and causes a disease. So the other part of this is what you might want to ask Ben about was, well, what is the signal for the spore to know it's in the gut, and when it's right to turn into the cell, the vegetative cell, as it's called, the pathogenic cell.
That's when it produces the toxins.
And that's because in the spore it's inactive. It does nothing. It just sits there.
Yeah.
And we know spores are great at sitting and doing nothing.
Super good, right?
Very good. And so what we found, the other part of the story was that the other feature that the gut does very well is break down bile acids. So the gut bacteria break down your bile acids. You produce these complex bile acids. The gut bacteria break them down for whatever reason, because some of them are antimicrobial, so it's a protection mechanism. And in doing so, it breaks down something called TCA, taurocolic acid. And it takes this molecule, snips it in half, and when it does so, it now removes the signal for the spore, because TCA is what the spore senses. The spore senses high levels of TCA, taurocolic acid. When it senses that, it knows it's in the gut and it starts germinating. You reduce the taurocolic acid, which what happens in a healthy gut, because the bacteria break taurocolic acid down, remove that signal, spores don't germinate. And so what FMT, IMT was doing was putting the bacteria back in again that could take that TCA signal, degrade it, and remove the signal from the spore. And so the spores weren't germinating. So with Valerate, we could actually kill the pathogenic cell, stop it from growing. With the TCA removal bacteria, we could remove that signal. So now what we had was spores weren't turning into vegetative cells, and any vegetative cells that were there were being affected by the Valerate.
Wow, perfect.
And so we had this sort of double whammy on its life cycle. And so that's what we're taking forward now, and trying to see if we can turn that into something that's a drug.
Absolutely.
Because it removes the whole need then for finding donors and C. diff, and actually it's safer.
Just to push back a little bit, do you not think you might need the bugs in FMT, IMT to confer more general colonization resistance? Like there might be more to it, or do you think that a combination of the IMT and the Valerate and the BSH could be the ultimate?
I think if we can remove the C. diff and the toxin, the gut microbiota will recover.
Oh, interesting.
I think that's one of our feelings, because after first case of C. diff and you give an antibiotic, the gut microbiota starts coming back until they relapse again.
Yeah.
So we think, ideally what it might be, but you're right, it might be that you need a seeding event.
You need a reset,
and you need these other things just to really give it that boost.
Fascinating. So are you and your team working on sort of similar workflows and other indications?
So you're right. So we're also using FMT, IMT to control antibiotic-resistant bacteria in the guts of patients, renal patients, urological patients, and hematological patients.
Wow.
So these are all patients who we know have lots and lots of courses of antibiotics.
Yeah.
Because either they get urinary tract infections. For the urological and renal patients, they just get persistent, and then after a while, they've had so many antibiotics that they're colonized by bacteria, usually in their gut, which are resistant to all the antibiotics. So we need to control that.
Right.
And then in the hematological patients, people with blood cancers, because their blood doesn't work as well, they get lots of bloodstream infections, so they get lots of antibiotics, and they get to stages where they're in a situation where they're full of antibiotic resistance in their gut. And what we found is an IMT or FNT can modulate the gut bacteria to such an extent that we can really reduce the burden of that, and then sometimes just clear it out.
Amazing.
And so what we're doing there now is actually trying to apply the same sort of process to ask a question, well, what is happening? Why is that? Why is FMT doing that? Is it metabolite driven, or is it immunological driven?
Right.
Or is it gut barrier driven? Because all these different things are features that could control movement of a bacterium from the gut into the host.
Right. Right, Julian, we are low on time now.
Oh,
we're pretty much pretty much there, right?
Yeah,
we're done. So we should
wrap it up.
We should wrap it up, but we should get you on again to just push more and more on this reverse engineering FMT,
I'd be glad to do
IMT,
it.
and other diseases, because you've been a lot of fun and a wealth of knowledge. So thank you very much.
Thank you very much, James.
Cheers. To learn more about the guest on today's episode, or more about the Inside Matters podcast, please visit insidematters.health. Thank you.